Okay, class, we ended last session talking about implantation, and now we're on the formation of the placenta, placentation. Okay, basically the placenta is formed from the mother's tissue and the conceptus tissue. Okay, the embryonic trophoblastic tissue and maternal endometrial tissue. The chorion, remember the chorion, that's this material here. The chorion becomes vascularized. Okay, it extends to the embryo as umbilical arteries and veins and lie immersed in maternal blood. You had the decidua bastalis, which is this area here, and then the capsularis was here. Okay, so the decidua bastalis is the one that's going to be more involved. That's the mother's part, involved in the formation of the placenta from the mother's end. The decidua capsularis is this outer area here. It's not involved in that. Okay. Now, the embryonic placenta barriers include the chorionic villi, the endometrium, and so forth. So we're going to go to this other here. Go to this. And here's the placenta right there. Here's the decidua bastalis. Decidua bastalis is the mother's part. Here's the chorion frondosum. That's the portion of the chorion that we'll say combines to form the placenta with the mother's tissue. Okay. Here is a look at a placenta. This is the fetal side. That's your umbilical cord. And here's the maternal side. We call them cotyledons. These bumpy areas here. That's the maternal side. Okay. So right here, what they're trying to show in this drawing, right here, is this part right here. And here are those cotyledons there that you see, because here's the umbilical cord coming there. Umbilical cord to kind of give you, and you can look at this later. All right, so we go a little further here. So now if I look at the layers of the placenta, starting with the uterus to the fetus, I have the decidua, which is the mother's part, transform endometrial. Then I have something called this layer, fibrinoid. Then I have trophoblast, that's kid, covering the gaps and growing into the walls. Then I have some blood-filled spaces. And then we have the syncytial trophoblast, which is the portion of the trophoblast that contributed to the placenta. Okay. Here's some pictures that you can look at later that show the layers of the placenta there. Okay. All right, I'm going to come over here now. So here again is another picture that you can glance at, and it just takes time to kind of lay out everything. Come here. Here again is that decidua, so you can see a good picture. Decidua bastalis, that area that of the mother's part that contributes. Here are those spiral arteries and so forth like that. All of this here that contributes to that particular aspect. Come over here, see if I see anything that I want to pick up, and I don't think so. Okay. So I'm going to come back here now. We'll go a little further. There again is the placenta. Right there. So now we're coming to germ layers, and I'm going to come back here. Which we talked about everything here. Decidua bastalis. So nothing to cover there. Okay, except here's the chorion frondosum, that portion I mentioned of the chorion that contributes to the placenta. Now let me just mention this. Since part of the placenta is developed from the conceptus, and part of the conceptus genetic material is the dad's genetic material, then essentially this is really somewhat foreign. Okay, the placenta of the kid too, basically, to the mother because there's other genetic material. So we're trying to find out why it's immunologically privileged, that it's not being attacked by the mother's immune system. And it appears that some T regulatory cells, these are lymphocytes, T lymphocytes, produced outside the thymus protect these paternal antigens. So in other words, there's several theories of this, shall I say. And then there are some special natural killer cells that seem to remodel the spiral arteries in the stratum function. Now, all of this to try to protect the mother from attacking the offspring. So I'm going to bring that up. All right, I come over here, and now we're looking at these germ layers. Germ layer stands for germination. Germ layer, there are 3 germ layers, as we'll discuss, that you should have discussed in ANP1 also. Endoderm, mesoderm, and ectoderm, germination layers. Okay, so the blastocyst develops into a gastrula. The gastrulation is this process of forming the 3 germ layers, ectoderm, endoderm. Before becoming a 3-layer, the inner cell mass subdivides into an upper epitoblast and a lower hypoblast. These layers form 4 embryonic membranes. Okay, so we'll come further. The amnion epiblast cells from the transparent filled with amniotic fluid. Now the amnion is going to form between the inner cell mass, shall we say. The amnion is going to form, let me go back here, between the inner cell mass, and I'm going to come back here. So at the trophoblast, the inner cell mass forms 2 germ layers. And the amnion, the 2 germ layers are the endoderm, will form the gut, and ectoderm. The amnion forms from the fusion of the germination layers, these 2, and forms a sac around the embryo. Now I'm going to go here. That's where that amniotic fluid would be. I'm trying to see if I can't find a picture here. So here's that amnion right there. And here is the embryonic disc right there. And it's starting to separate to give this amniotic cavity right there. Right there. That's what we're looking at there. Okay. So this is what we're seeing. Okay. So I'm going to go further now. So other extraembryonic structures of the yolk sac, and lantois, and the chorion, which we talked about, that helps form the placenta. Okay. Gastrulation is the process of forming these germ layers. So during the 3rd week, the 2-layered embryonic disc becomes a 3-layered embryo. The primary germ layers are ectoderm, mesoderm, and endoderm, but ectoderm and endoderm form first, and then later comes the mesoderm. And this process has to do with formulation of a primitive streak, but we won't get into that. That's getting more into a course in embryology. But just know that 3 germ layers will form ultimately. And each germ layer, germination layer, forms different structures in the adult. Like here, endoderm, mesoderm. Mesoderm forms all your connective tissues. Endoderm can form the gut and so forth. Ectoderm forms the nervous system and part of the epidermis, or at least the epidermis, shall I say. Here are these germ layers coming in. Right there. Okay. So where we go now. Okay, class, we talked about the formation of that amniotic sac. So let's talk about this amniocentesis. Well, an amniocentesis basically is it's a test where a needle is stuck through the maternal abdomen into the amniotic sac to try to obtain fluid to do genetic testing and other things, particularly like for Down syndrome, because remember the amniotic fluid, or shall I say the amniotic fluid, is a product of the conceptus. Initially, it's a secretion off the wall of the chorionic area, and later, the amnion, and later it'll be basically being secreted by urination of the fetus, of the embryo inside. Okay. So samples from the amniotic fluid can help diagnose genetic abnormalities such as Down syndrome. Another test that can be done is to get a pinch off of the chorionic villi. Now, this test here, the amniocentesis, you have to have sufficient time to have a space between the developing embryo and, or shall I say, developing fetus. Developing fetus, embryonic ends at 8 weeks, and the sac itself, amniotic sac, so that you don't poke the fetus with the needle. But an alternative test is to take little pinches off the chorionic villi, which would be more or less within the placenta underneath, and that can be done earlier. Another thing can be done are blood samples from the mother, because some of the blood has leaked into the maternal circulation, and we know that now, even though it's supposed to not be an exchange between the mother and the fetus, you can take some samples from the maternal blood. Here's a picture of an amniocentesis being done there, drawing some fluid. Now, the next thing we go to is molecule exchange across the placenta. Maternal blood volume increases by 40% and total peripheral resistance. You're increasing this blood volume in order to have sufficient blood volume for exchange, not between the mother and the fetus, but in order to perfuse the area of the placenta, but not to send, there should be no exchange between mother and fetus. So you increase this blood volume in order to have enough to perfuse into the placenta area. And you also drop total peripheral resistance. We talked about that before, in order to be able to, some more vasodilation to be able to do this. In the umbilical cord, you have 2 umbilical arteries and you have 1 vein generally. Okay? And so this is what we have in this exchange. So the mother basically is sending oxygenated and nourished blood through these vessels on the underside of the placenta. And so let's go to that. Let's take a look at this now. All right. Here is the maternal artery sending blood in to these villus spaces, these spaces. It is oxygenated and nourished. Okay? Maternal arteries. Now, in the umbilical cord, here's the umbilical cord here. Right here. Here is, and coming in, here's the umbilical artery. Should be 2 arteries and an umbilical vein. So the kid is sending blood this way and then picking up an exchange. So his blood is deoxygenated, okay? But however, because it's coming from the kid and leaving the heart, it's an umbilical artery, but it's deoxygenated. And then it will pick up oxygen and come back in the umbilical vein area and go to the kid.